Okay, what we're talking about today is non-Mendelian genetics. So there's not going to be an exact dominant or an exact recessive. We're going to have other things going on. So these are the exceptions to Mendel's rule. So it's called non-Mendelian genetics. The first exception to the rule is called incomplete dominance. This is where one allele is not completely dominant over another. The heterozygous phenotype is going to be somewhere in between the two homozygous phenotypes. So the classic example is you have a red flower that you write RR, and you have a white flower that you write WW because neither one of them is dominant over the other. When you combine them, when you crossbreed them, you get a pink flower, so it's in between the red and the white. You don't write PP for that. You write one R and one W because you're combining the red trait and the white trait. You're blending those together. So in this example, it says 4 o'clock flowers. The flower color is due to incomplete dominance. So some are red, some are white, others are pink. And it wants us to cross a white flower. So I'm going to put WW for white with a pink flower. So I'm going to put RW for the pink. I'm going to cross those. My genotypic ratio is going to be 2 RWs to 2 WWs. Don't get confused about genotypic and phenotypic. And my phenotypic ratio is going to be 2 pinks to 2 whites. So that's incomplete dominance. You have a blending of traits. Another exception to Mendel's rules is called co-dominance. There's a subtle difference here. Instead of there being a blending, both alleles contribute to the phenotype. So both are seen together. So in this example, I have a black rooster and a white chicken. So it's BB and WW. I don't get a gray chicken. I get a black and white chicken. So I have some feathers that are black, some that are white. They may look gray. So like, look at this chicken right here. He looks kind of gray, but if I took a magnifying glass and looked at his feathers, I would see some are black and some are white. They're not gray. So in this example, it says in chickens, BB calls for a black chicken, WW calls for a white chicken, and BW calls for a blue Andalusian chicken. And it wants us to cross a black rooster with a blue chicken. So they call it blue because if you look at it from afar, it can kind of look gray-blue. So don't let that confuse you. But that means they've got black and white feathers. Okay, so we want to cross a black rooster, so that's going to be BB, with a blue chicken, so that's going to be BW. Okay, so I have a black crossed with a blue. I'm going to cross those. I get, again, 2 BB to 2 BW or 2 black to 2 blue. So very similar to incomplete. There's just that subtle difference where you're not blending them. They're both showing at the same time. Okay, next exception to Mendel's rules, multiple alleles. So this gets a little bit more tricky. There's more than 2 possible alleles in the population. So here I've got 4 different alleles. I've got big C, CCH, CH, and then plain little c. So there's going to be more than 2 or even more than 3 possible phenotypes. Here I've got 4 possible phenotypes. So my 4 alleles are big C, which is the wild type or brown. That's the phenotype. I've got little c with a CH. That's chinchilla, which means if I looked at the fur, the bottom of each piece of fur, each little hair would be white, and the top of each little hair would be black. They look kind of gray. I have C with a little h. That's Himalayan. So it's going to be mainly a white rabbit, but it's going to have black extremities. And then I have just a little c, which is albino. So it's white fur with red eyes. So the interesting thing about this is if I look at these from albino, going back up here to brown, they're getting increasingly dominant. So brown is dominant over all 3 of these. Chinchilla is dominant over Himalayan and albino. Himalayan is dominant over albino, and albino is recessive to everything. So in our example, they want us to cross a CHC with a CCHC. So this one right here, that CHC, would look Himalayan because Himalayan is dominant over albino. And this one right here would look chinchilla. The CCHC would look chinchilla because chinchilla is dominant over albino. So I just set up my Punnett square just the same. I just have these little superscripts up here. So I have a CHC, and then I have a CCHC. Okay, so when I cross these, I'm going to put the CH first because it's dominant. CCH with CH, and then here I'm going to put CCHC, CHC, CC. Okay, so what will this one look like? Well, chinchilla is dominant over Himalayan, so it's going to be chinchilla. What would this one look like? Chinchilla is dominant over albino, so it looks chinchilla. This one right here, Himalayan, is dominant over albino, so it looks Himalayan. And then this one, I have two little c's, so that's albino. So my phenotypic, we don't really need to worry about genotypic ratio, but phenotypic ratio is 2 chinchillas to 1 Himalayan to 1 albino. Okay, so that's multiple alleles. All right, the next thing we need to look at is blood type. Blood type brings into play multiple alleles and codominance. And I've also linked a video from the Amoeba Sisters about blood type, and I really recommend you watch it because it really makes blood type clear. But there's 4 blood types, A, B, AB, and O. So my multiple alleles, I have an A, a B, and an O. Those are my multiple alleles. I also have a co-dominance thing going on because A and B are equally dominant. They don't dominate each other. So that's why I can be type AB because they're both show together. Okay, so here's my A blood cell. It's got these little antigens on the outside that are A. Here's my B blood cell. It's got these antigens that are B. The AB blood type has A and B antigens showing together. And then the type O has no antigens. Okay, so that's what that's referring to. Okay, so to be type A, when I write the alleles, I write them over an I. You don't have to understand why. It stands for these antigens, okay? So I either have an A, a B, or the little I stands for an O. So to be type A, I can be AA because the A is dominant, or I can be AO because the O is recessive. To be type B, I can be BB, or I can be BO, and I just write that with a little I. To be type AB, the only genotype I can be is A and a B. I have to get an A from one parent and a B from another parent. And then to be type O, I have to get an O and an O from both parents. So I have to be OO, which you write little I, little I. Okay, so the O is recessive, A and B are equally dominant. Okay, so in this example, it says a wealthy elderly couple dies together in an accident. Soon after, a man shows up to claim their fortune, claiming that he is really their only son, who ran away from home when he was a boy. Hospital records show the deceased couple were blood types AB and O. The claimant is O. Was he an imposter? So I just have to set up a Punnett square. I'm going to drop those I's, and I'm just going to write A's, B's, and O's to make it a little bit easier. So up here, I'm going to put this parent that's AB. And then on the side, I'm going to put this parent that was O. So remember, O is recessive. To be O, they have to be OO. And then I'm going to do my cross. Okay, this offspring right here is type A, type B, type A, type B. And the claimant was type O. So could he have been their child? Was he an imposter? Well, yes, he was an imposter because there's no way this couple could have a child with type O blood. Now, pretend if the imposter or if the claimant had been type A and we got this, that would not be proof he was their child, but it would at least not rule him out. So when they use blood typing in criminal cases, mainly it's to rule someone out. It's not to say they're the exact one. So we can rule out anybody that's type O blood or that's type AB blood. But if they have type A blood or type B blood, they're just one of many, many, many people that could be that child. Okay, sex-linked traits. So remember, a female has XX chromosomes for their sex chromosomes, and a male has XY. Any genes that are on these chromosomes are called sex-linked genes because they're on the sex chromosomes, sorry. Since most of these alleles are carried on the X chromosome, they're going to have a different effect on males than females. What that means is males are going to be more likely to get it because they only have to get it one time to have it, whereas females have to get it twice because they have two X's. Some of the common sex-linked disorders that you hear about are color blindness, hemophilia, and muscular dystrophy. Okay, so the allele for color blindness is carried on the X chromosome and is recessive. A man whose father was colorblind has a colorblind daughter. So here's his daughter. She's got X with the recessive colorblind gene, X with the recessive. She has to have it twice because it's recessive. Okay, and it wants to know, does the father have to be colorblind? So we just need to put a little Punnett square. This is the daughter. Okay, she had to get one XC from her mom and one XC from her dad. She had to get it from both parents in order to have color blindness. Now, up here's dad. What's his other sex chromosome? Men are XY, so that's his other sex chromosome. Okay, so he has XCY. And the mom, she's XX. We don't know what her other X is. She could be normal or she could be colorblind. Okay, so did the dad have to be colorblind? Yes, because he only has this one X. So if he gave an X with a colorblind gene to his daughter, he's colorblind. Now the mom could be normal and still pass it on to her daughter. Okay, look at the son right here. Where did he get his colorblindness from? Well, he got the Y from dad, so he got the colorblind gene from mom. So yes, the man has to be colorblind because he had to pass the trait on to his daughter. Okay, last thing I'm just going to mention, just so you've heard the term, poly means many, genic, referring to genes. So many gene traits. These are traits that are determined by many genes. So skin color is the perfect example of this. There's many genes contributing to skin color, which is why we get such a wide spectrum of skin color. You can have really, really light skin, really, really dark skin, or anywhere in between. Okay. We're not really going to do Punnett squares or anything or deal with those, but just know that there are some traits like skin color, eye color, hair color that are controlled by many, many genes, and those are called polygenic traits. The end.